Hello everyone, welcome to the instruction video before the Metagenomics Lab course. My name is Wenjing Meng, a PhD student in MPAC group. This video is going to help you prepare for the lab exercise, including downloading the bacterial samples we are going to analyze, applying for a CSC account, as well as computing resource on Puhti. The very first thing is, go to this Google Drive link and download all the files in this folder, which we will upload onto the Puhti. So let's do this together. Copy the link, paste it here and enter. And you could see 6 files in total, 4 zipped sequence files ending with fastqgz, 1 grouping text file called op-metadata, and 1 text file named op-config. Let's take a look at the content of the sample grouping file. You could see much grouping information prepared manually about these 2 samples, namely allsoil.1s and allsoil.2s. Then, these 4 fastq.gz files are basically the forward sequences and reverse sequences generated from 2 soil samples we just looked at. The last file is this op-config text file. Let's open it, and you can see it indicates where these sequences will be located on the Puhti computing platform. So here is an important issue that the corresponding pathname must be corrected to your own pathname once these files are uploaded on Puhti. In this way, could the system recognize where your sequencing files are? So select all the files and download them. So you can get all the files you need for the analyze. The second part is to register in CSC. So go to this link, click Login or Register here, select University of Helsinki, log in with your own university username and password. And then you come to this web. Fill the form with personal details. Choose Research or Study for the primary purpose part, and finish the rest part. At last, click these terms and click Submit. So you will see this registration successful note at last. Then it will take a few minutes before you are able to log in. After a while, you could log in CSC on the same website where you applied for your new account. Here I have already logged in with my own account, and I'm going to show you how to create a new project. First, click this Projects button on the left panel, and then click this Plus New Project. You could use random project name and description to fill this form. For my case, I want to use Metagenomics Lab course for the project name and microbiome analysis through Chimp2 platform as description. Now choose course for the project category. And then you could see more options. We must fill the necessary areas. So I'll choose today as a course start date and maybe October 24th as the end date. And then for the project resources, we must fill all these areas. And you could fill the fill them same as mine. At last, I must click this Terms of Use. And finally, I click this Create Project button. Yeah, and then it shows the project was successfully created. And it shows right here. So now you can see all the information about this project. And our next step is to apply the computing resources, which is Puhti in our case. So we can either choose add services from here or add services from the services area. And we can choose Puhti, which is a supercomputer for our resource. And we click this Add Selected Services. And now Puhti has been enabled for our project, and it will take up to 30 to 60 minutes for the changes to take effect. So we must wait for their activation. It's an hour, so it should be fine now. Let's log into Puhti by clicking this login button. And again log in. Choose Haga login. So choose this Compute Not Shell button. Choose the right project name. Change the local disk to 10, but keep other default settings. And then click Launch. You'll see this session is running. Wait a few seconds. Now it's ready. So we choose Connect to SSH session. And now you can see the working environment is loaded. Let's close this and go back to Puhti homepage. Then click the Home directory. Now we should upload all the files we have downloaded from that Google Drive link. So click Upload, Browse Files, choose all the files, Open, upload 6 files. And now we have all the 6 files we needed for the analyze. And here the important thing is to modify the file path information in this opconfig text file so that it suits your own case. Let's do it on our own computer file and then upload it. So click this op-config text file on your own computer file. And in my case, my path should be users wenjmon, and I delete this for information. And I do the same for the next 3 files. Yes. Save. And then I delete this config file and re-upload the new config file. So now all the files are ready. Let's click this Open in Terminal button. So this working page is back again. Let's input the command ls and enter. It will show all the existing files in our directory. And then input the command module load gem2 and enter. So it will show that environmental loaded. And now you are ready to go.